Thank you to the second speaker, Heather Bracken-Grissom, who's going to talk to us about the evolution of vision and bioluminescence. So over to you, Heather.
Thank you so much. Okay. Let's get started. There we go. Can everyone see this all right?
Yep, that's fine. Thank you.
Okay, perfect. Awesome. So yeah, I just wanted to start off by also thanking everyone for coming today and allowing me to speak in this fun webinar. And I'm going to be talking about the evolution of light organs and vision in deep sea shrimp. And this is something that my lab has been studying for quite some time, about 9 years now. And just a little introduction to myself. My name is Heather Bracken-Grissom. I am currently the Assistant Director of the Coastlines and Oceans Division and also an Associate Professor of Biology at Florida International University. So I'm located in Miami, and this is just a picture of our campus. So I'm on the Biscayne Bay campus in South Florida. And I did need to just show a picture of my lab. We call ourselves the Cristomics Lab, and I'm going to get to that in just a second. But I did want to show the faces behind a lot of the research that I'm going to talk about today. So what do we study? So you might kind of infer from the name, we study everything crustaceans. So we combine genomics and taxonomy to study everything about crustacean biology. We're particularly fascinated by biodiversity, and we've done a lot on biodiversity. I'm not going to talk too much about that today, but this is really one of the pillars of my lab, is describing and documenting biodiversity from the mesopelagic. We also are an evolutionary biology lab, so we like to study evolutions, how these animals are related to one another using genetics and genomics. And then adaptation. So we're particularly fascinated by bioluminescence. If we combine these three pillars together, it allows us to tell something about the patterns and processes which are happening in the oceans. So I mentioned previously that we've done a lot of biodiversity research. So this is a picture, these are pictures of animals that we've taken on our subsequent cruises. We've spent about 200 days at sea since 2015, and we've been able to document a lot of the beautiful biodiversity from about 0 to 200 meters in the Gulf of Mexico. So we're using traditional taxonomy, and we're combining this with DNA barcoding and genomic methods to characterize biodiversity. And if any of you are interested in these publications, I'd be happy to send them your way. They're also on my website. But what I wanted to talk to you about today was this fascinating world of bioluminescence and vision. So we all know that marine bioluminescence is extremely common in the deep sea. There's over 700 genera that are bioluminescence, and 80 of those are marine. So I don't really need to, tell you too much about this phenomenon in the twilight zone, but I did want to share some pictures from some recent expeditions in the northern Gulf of Mexico. So on the top, you see the deep sea coral Iridogorgia, and then the bioluminescent signature. Again, a deep sea coral Paramaricia, a beautiful sea anemone, and also this sea pen. But the animals that really fascinate me the most are these deep sea bioluminescent shrimp. And when you think about bioluminescence, a lot of times people don't think of decapods. But deep sea shrimp actually have these remarkable different modes of bioluminescence. And this is something that my lab is particularly excited about. So this first mode is this bioluminescent smoke screen, and they use this as a form of defense against predation. There's a lot of other animals that use this smoke screen when they get startled. And we were able to actually get a video of this on one of our recent cruises. So this is what it actually looks like. This animal is in a V tank, so it can't escape. If it was out, in the open ocean, it would be tail flipping and escaping at the same time. But all of this was just videoed, recorded in a dark room. And this is the color of bioluminescent. The shrimp is Oplophorus gracilarostris. So we know that they use this as a form of defense. What we've been studying is these bioluminescent light organs, and deep sea shrimp have a remarkable diversity of bioluminescent light organs. This is just one family called the Sergestidae, and they contain 4 different types of light organs. The first are these unlensed photophores. These photophores are all along the carapace of the shrimp, along the tail fan and the antennal scale. You have lensed photophores, and if you look really close, you can see there's these bubbles that come off of the photophore. So these lensed photophores, and then there's these internal photophores called organs of Pesta. The 4th type within the family is actually a complete lack of photophore. And so there's 4 types just within this one family, and we have studied the diversification of these light organs using genetics and phylogenetics. I'm not going to be speaking too much about that today, but we are really interested in how these light organs evolved. We know that these light organs function in a deep sea game of hide and glow called counterillumination, which I'm sure you're all familiar with, so I'm not going to get too much into it. But a fun fact is that counterillumination has only been experimentally shown a handful of times. And one of those times was actually in this family of deep sea shrimp called Sergestids. So Sergestes similis, this is the publication from Warner, Latz, and Case. They've actually shown counterillumination. The first publication was in Science in 1979, but they've also been able to manipulate these shrimp in the lab since that publication to do various experimental studies with counterillumination and Sergestids. What you see here on the left-hand side is just a cookie cutter shark that is counterilluminating. So we know that counterillumination breaks the silhouette, and it's a form of camouflage in a lot of these animals that live in the deep sea. So what my lab's been really interested in over the past, I would say 8 or 9 years now, is do these light organs actually have an expanded role within or beyond this game of hide and glow, so this game of counterillumination. So it's documented that counterillumination is common, but we want to know if these photophores actually can play expanded roles or within counterillumination. So the 2 case studies I'm going to talk to you briefly about are studies that my lab has done. One is this light sensor study, and second is we're looking at the role of photophores in communication. So the idea that these photophores, so I was, I've collected hundreds and thousands of these deep sea shrimp, and I've looked at the patterns, and I started to ask myself about the role in counterillumination. So most of the time, these photophores are in fact, heavily along the ventral surface and the lateral surface, which we would all expect that they would play a role in counterillumination. But if you look really closely at these photophores, they're not just along the ventral and lateral surface of the shrimp. They're actually covering the entire body. So you can find them on the dorsal surface, and you can find very conspicuous streaks of photophores along the eyes, on the very top of the eyes, along the eye stalk. So it made me kind of ask this question: are they possibly playing more of a role or an expanded role within counterillumination? And specifically, we wanted to ask this question: can photophores not only emit light, but can they also detect it? And this is the idea of extraocular photosensitivity. And this isn't a new idea in science. It's actually been documented quite a few times in the marine realm. So this term extraocular photosensitivity is the ability to detect and respond to light outside of the eyes. So it's been shown in the 2 feet of echinoderms, the central nervous system of arthropods, the dermal chromatophores of cephalopods. But the study that really inspired me was that this extraocular photosensitivity was documented in the light organs of bobtail squid. So we went out and we looked at these photophores of these deep sea shrimp in a little bit more detail to in fact ask if those photophores could actually not only emit light, but can they also detect it? And what we found was a study that we published in 2020 in Scientific Reports, where we used a combination of techniques. So the first thing that we did was we used genomics, something called RNA-seq, and we extracted the RNA out of these photophores. We also extracted the RNA out of the eyes to look at gene expression. And we found that the photophores contained the same genes that were responsible for light detection that the eyes contained. So the photophores not only contained opsins, which are genes responsible for the detection of light, they also had genes that were involved in all aspects of phototransduction. So we were able to identify these genes in the photophores. We also did shipboard experiments, which is shown right here, where we asked if there was a cellular response when these photophores were exposed to light versus when they weren't. And we did see several different responses at a cellular level. And then we used molecular techniques called in situ hybridization and immunohistochemistry. And we wanted to see where those genes were actually expressed in the photophore and if we could actually label the protein. So we used a combination of an integrative approach of genetics, molecular, and physiology to examine photophore light sensitivity. And we did in fact find mounting evidence that these photophores are photosensitive. So the next real question was, well, what are they using this for? What are they using these photophores to detect light? And that's still something we don't know. My lab's interested in DVM, diel vertical migration, and could this be a trigger for DVM? We know that light does control some aspects of vertical migration. Could photophores actually be playing an expanded role in DVM? We do think that these photophores are allowing, one of the things, let me back up really quickly. One of the things that we still don't know is how animals precisely match that of downwelling sunlight. So these animals can precisely match that of downwelling sunlight so that they can match the ambient light, but the mechanism is still unknown. So we do think that these photophores may be working in conjunction with the eyes to help regulate counterillumination. So that's something that's still an interesting question. And if you know any of the photophore literature, there's also a really cool hypothesis about these eye-facing photophores in fish that are thought to help modulate counterillumination. So the last thing I'm going to be talking about quickly is that we're also interested in the role of photophores in communication. So, you know, I think it's very plausible that we don't know a lot about bioluminescence, and we wanted to know if these photophores might also be playing a role in communication. So I showed you the diversity of light organs that this one family of surgestids have earlier, but another remarkable thing is they don't only have this remarkable diversity of light organ type, they also have species-specific diversity in light organ arrangement or pattern. So what you're looking at here in this figure are these internal organs of Pesta. So this is a surgestid shrimp. This is Diosurgestes hanseni. And I dissected out that posterior organ of Pesta so that you can see that light organ, the pattern of those light organs. And if you look at figure A, each of those are a different species of surgestids. So light organ pattern differs significantly across species. They have conspecific patterns. If we look at the Surgia species, so if you're familiar with surgestids, they've traditionally been classified based on these internal photophores versus these external photophores. But these Surgia representatives, they also are species specific. So what you're looking at here are the patterns of photophores along the antennal scale and the uropod. So I actually use these patterns to identify these shrimp in the field, and they're pretty good indicators of what species you have. So you can use photophore patterns to identify down to species, and taxonomists have done this for a long time. And there is sexual dimorphism reported for several of these species. So it kind of lends you to ask the question, if these organ patterns may play a role in species recognition. So we chose 3 very different species patterns to take a look at this question. 3 species from the Sergestes group. And we did various modeling and visual acuity tests. So I'm not gonna get into too much of the details, but this work was published also in 2020 by my postdoc, Schweiker et al. And we asked questions like, how far can these deep sea shrimp actually detect bioluminescence? So we measured various aspects of their eyes and visual properties. We modeled the bioluminescent signal. And we also asked if they could actually see these organ patterns. So we used a program called visual acuity to see if these animals actually had the resolution to be able to discern these different patterns from one another. So we found a few maybe not so surprising things. The first thing that was pretty interesting was that we were able to predict that these maximum sighting distances ranges from about 8 to 20 feet in this group. So we know that these shrimp swarm. They make up a lot of that deep scattering layer that Matthias was talking about earlier. So they do swarm, and they're found in very high densities. So, you know, this is a range that they can actually detect the bioluminescent signal from about 8 to 20 feet away. Now, whether they could detect patterns or not was a very hard no. And what I'm going to show you is all of these blurry images is how they see their own patterns and the patterns of the other species that we modeled. So we came to the conclusion that they cannot resolve patterns, but they can detect bioluminescent signals over a relevant distance. So the very last thing I have was that this paper is just going to come out, I believe, in the next 2 weeks. And this is pretty exciting because the next thing that we did is that we took, in a follow-up study, we examined the ecological predictors of eye size evolution. So taking into consideration phylogenetic history. So we tested several ecological factors. We tested depth, diel vertical migration range, habitat, light organ type, all on eye size evolution. And the only thing that was a significant predictor of eye size was light organ type. And so this was really exciting to us. And this suggests that light organs may play a larger role in conspecific signaling than we previously thought. So we're moving this into the lab. We're going to try to do some behavioral studies with deep sea shrimp, which is probably going to be extremely challenging and maybe not so smart idea, but we'll see if we can get some of this to work. So in conclusion, we basically, some take-home messages is that we still really have a lot to learn about bioluminescence. We have mounting evidence for light sensitivity in photophores and deep sea shrimp. We don't know how they're using this photosensitivity. This is a direction we really want to go. And there's a possible role that these deep sea shrimp could be using these light organs for communication. And with that, a quick acknowledgement to my funding sources, and also a quick plug that we are looking for collaborators. So a whole nother aim in my lab is we study deep sea population connectivity. So we use population genomics to see how populations, global populations are connected to one another. If you're going out in cruises in the Pacific or the Indian Ocean specifically, or the Atlantic, we'd really love to hear from you and possibly collaborate on deep sea population connectivity in the future. So definitely give me a shout out. My email is hbracken@fiu.edu. I'd love to hear from you. And with that, I'll take any questions. Thank you very much.
Thank you, Heather. Absolutely incredible stuff. Thank you. Do we have any questions for Heather? Let me just check the chat in case we've got anything there. If anybody has any questions, feel free to raise their hand. So I've got one just to get things started. I've got several, but I'll limit myself to one. So if they're using bioluminescence to signaling, I guess my question would be, what are they using that signaling for? Because it must come as a risk as well. You know, if you've got a dense aggregation of these and they all start flashing, then isn't that just like ringing the dinner bell for, well, for the taupe sharks or, you know, anything else which is in the area?
Yeah, absolutely. I mean, there's immense trade-offs. I do think, you know, they do mate, like they mate by copulation. So they do have to find each other to reproduce. So I think if anything, they might be using bioluminescent signaling to find each other for mate recognition, but that's definitely not all of it. I mean, it wouldn't be enough, right? So I think olfaction and chemical stimulus is also plays a huge role. And I think there's a lot of really interesting things that can be looked at there. These surgestids have these remarkably long antennae that are sometimes 10 times the length of the body. I mean, it's one of the characteristics that you see and how you can identify a surgestid shrimp from like ROV footage. They're pretty indistinguishable by the length of their antenna. So I think they might be using the signaling to for mate recognition, but then following up with like different chemo olfaction.
Fascinating. Yeah, so I could add other questions, but I should see if anybody else would like to ask anything. Is there anybody out there? Anybody else who's got any sort of questions before I sort of carry on? I mean, I guess the other thing I've got is, I mean, is it energetically expensive to do this for an organism? I mean,
I'm not sure how expensive the bioluminescence is, but I mean, they are. One thing I didn't talk about was just DVM in general, and how expensive that must be. But one thing we've looked at is the evolution, the diversification of these light organs across, plotted against their DVM behavior. So these shrimp, although they all participate in diel vertical migration, they're migrating at different extremes. So a lot of them migrate a lot shallower than others. Some of them have a lot more narrow range of DVM, and it appears that their light organ diversification has evolved based on that migration and the light environment in which they occupy. So for example, some of the surgestids that lack photophores altogether, they don't vertically migrate into extremely shallow waters where they would need photophores for counterillumination. So we've been able to plot organ diversification across against DVM behavior, and we're starting to see some really interesting patterns emerge. Those lens photophores only appear in animals that are associated with the benthopelagic. So why would lens photophores only be associated with benthopelagic species? So there's some really intricate and complex things going on here that I'm not really sure about. I don't think bioluminescence is necessarily energetically expensive, but eye investment is. So that's why we're really interested in eye investment related to bioluminescence also.
Excellent, thank you. I see Lenny's got a hand up. Lenny.
Yeah, hi Heather, thanks for the nice talk. So you explained that your modeling indicates that their visual capabilities are not highly resolved enough to distinguish species-specific patterns. And you mentioned they can regulate their photophore activity. So is there any evidence of species-specific signaling or like rhythms that they can signal, or has any of that been observed?
None of that has been looked at. So, I mean, the best examples that people have looked at these is in, you know, marine ostracods, sea fireflies, right? Where they perform these beautiful Morse codes to sexually signal each other. We have no idea. We do know that the photophores can be turned on and off very quickly. So they've actually been able to manipulate counterillumination in the lab, and it's a slow induction, which suggests hormonal control of the photophores. But as soon as they're turned on, it's a very quick response, which suggests neural control. So it seems like they might be under both hormonal and neural control. So they can turn on and off very quickly. They can control the duration, and they can move them, so the tilt. But no one knows anything about sexual signaling, and that's kind of what our next thing is to basically just count photophores. So we're going to collect all these animals and do a big monograph on photophore patterns and colors, because these photophores are also covered by different colored lenses. So some are these deep red, some are purple, some are deep blue. So the colors are different too. But I think it would be really just neat to look at sexual dimorphism. So how much sexual dimorphism actually exists in the patterns. And then that will lead you to a whole nother suite of questions. So.
Excellent. It looks like you have no shortage of questions yourself that you want to answer.
Yeah.
So to add to Heather's list of questions that she wants to ask herself, are there any other questions out there in the audience that would like to be put to Heather? No? Okay, so with thanks once again to both our speakers for really excellent talks, very stimulating. Thank you all for joining the call this month. We will have 2 more talks next month, but that will be run from the Australian time zone, so please